All right, this is chapter 15, Campbell's 7th edition biology, chromosomal basis of inheritance. So we're talking about genes and how they're inherited. We're moving on a little bit from Mendel. So a lot of this is based on Mendel. Researchers proposed and thought that potentially genes were located on chromosomes, and this fit Mendel's laws of segregation and independent assortment. And so the theory basically suggests that these genes have specific loci, which is just locations, and that they undergo segregation and independent assortment. And this is just reviewing from chapter 14. Mendel's laws on the left is segregation, 50-50 chance of splitting left to right, and independent assortment, one pair's splitting doesn't affect the other pair's splitting. Now, Thomas Hunt Morgan's experiments with fruit flies was the first evidence that chromosomes were located on Mendel's heritable factors, or on chromosomes, basically. That genes were where information was on chromosomes. So he used fruit flies. They breed really fast, every 2 weeks, and they have 4 pairs of chromosomes. So it's a pretty simple model. And he noted all the different mutations and wild-type populations. Here you see mutant phenotype on the right is white eyes, red eyes is the wild type, or normal. And so he took a P generation and he mated mutant white-eyed with males, and he mated with, them with female red eyes, and all the F1s had red eyes. So, red eyes must be dominant to white eyes. But then he crossed the F1s, and it showed a 3-to-1 red to white. But this time, instead of, as Mendel predicted, only the males had white eyes. And when he performed these crosses, this enabled him to identify that this was a sex-linked trait, and thus the gene was on the X chromosome. So here you see the white-eyed male, wild-type female going through. Here's the F1s, they're all red-eyed. And here's the F2s, you have only males as the white-eyed individuals. So again, first solid evidence indicating that a specific gene is associated with a specific chromosome, in this case the X chromosome. Now, the most complicated topic of chapter 15 is linkage, gene linkage. Everything that Mendel discussed and talked about were genes of various phenotypes that contributed to various phenotypes, and they were all on different chromosomes so that they assort independently. However, genes that are linked, meaning they're on the same chromosome and fairly close to one another, they assort together. The two genes go together to the gametes and to the offspring, which makes things a little bit more tricky, and Punnett squares get a little bit more complicated if you do them. Each chromosome has hundreds or thousands of genes, hundreds for the Y chromosome, very small, and then on the order of thousands for the X and other chromosomes. So he followed up his experiments by looking at how linkage affects the inheritance of 2 characteristics. So here we see his initial P generation cross. Here's the wild type, gray and normal wings. And on the right you see the mutant, black body and vestigial wings. And when he crossed them, all of them were wild type. But this time now they're heterozygous wild type. B plus B and VG plus V. VG stands for vestigial and B is black, of course, black body. And he did a test cross. He crossed it with a double homozygous recessive. And the result, what he found interestingly, was that the majority of the offspring were exactly like the F1 parents here. 965 of the, let's say approximately 2,500, were wild-type gray normal, just like this parent. B plus B, VG plus V, VG. And the same number, or approximately, were just like the other parent, black vestigial. And very, very small percentages were the combination of the two. So gray body, B plus B, with vestigial wings, VG, VG. And Mendel's laws in independent assortment and a normal dihybrid Punnett square doesn't predict this. It would predict equal numbers of all 4 phenotypes, but we didn't see this, or Morgan didn't see this. He saw very small numbers of these two. And he called these two recombinants. These two offspring, or phenotypes, are the result of crossing over, which occurs far less often than not crossing over, which is the how these two occurred, the non-crossovers. So genes that are close together on the same chromosome are linked, and they do not assort independently. Unlinked genes are either on separate chromosomes or are so far apart that they assort independently, meaning they're so far apart on the same chromosome that there's many, many locations where a crossing over event can occur, and they behave just as if they were on separate chromosomes. So here you see it, once again, the example, most offspring are like this, just like this parent, or like this, which was just like this parent. So this is typical Mendelian inheritance, where this is the different phenotypes of what you would see. But if we apply linkage to Mendelian genetics with Ps, and we look at color and round versus wrinkled, if we have a heterozygous parent, big Y, big R, little r, and we have we do a test cross, little y, little r, to form these gametes. These are all the potential offspring, and we would see in a recombinant, in a linkage problem, the majority of the offspring would be like this and like this. You'd have far larger percentages of these, and a very small percent would be these two. Mendelian inheritance predicts a 1 to 1 to 1 to 1, but if they are linked, then you get very small numbers of these, and we would call these recombinant offspring. And recombinant offspring are just those that show new combinations that are different from the parental traits. And when 50% of the offspring are recombinants, and that's how Mendel predicted when they assort independently, well, geneticists just describe it as a 50% frequency of recombination. Anything that is 50% and above simply means that they assort independently. It means that they're on different chromosomes, or that they're so far apart that they behave like they're on different chromosomes. So Morgan discovered that genes can be linked, as we previously mentioned, but sometimes it can be difficult to identify the linkage because they're the same types of phenotypes. So he proposed that some process must occasionally break the connection between the two, and this is what we now define as crossing over, and crossing over occurs between homologous chromosomes. So parent 1 donates chromosome number 1, parent dad donates chromosome number 1, and you have a homologous pair of chromosome number 1. If you crisscross and exchange DNA between those homologous pairs, that would be crossing over. Here we see the crossing over event right here during meiosis, where we've exchanged a little piece of the DNA, and as a result you see the little black line representing an allele that has switched over as well. And you can see that here in these two recombinants. So these two will appear in the offspring far less often than these two, because this event is less likely than this event. And the closer that these genes are together, the less often we'll see these recombinants. And you'll see that will become important when we do linkage problems later. Here's an example going through this of how a linkage problem is done. There's 391 recombinants between these two. You add them up and divide them by the total number of offspring. You're looking for the recombination frequency, so it's recombinants out of the total. Multiply it by 100 to get a percentage, and we have 17% recombination frequency, which for our purposes tells us that the genes, these two genes, B and VG+, are 17 map units apart from each other. So they're pretty close. They're 17 units apart from one another. If they were a bigger percentage, that would be because there were more recombinants, and thus there were more crossing over events between them. And we'll take a pause on this one.